Researchers use CRISPR to engineer a bacteriophage to deliver DNA into targeted members of microbial communities for precise genome editing. Bacteriophages or phages are viruses that infect bacteria. They are the most abundant biological entity and they can be found wherever bacteria live. To propagate themselves, phages inject their DNA into their bacterial hosts. Their DNA hijacks the host cell, forcing it to produce more copies of the phage until the cell bursts and releases thousands of new phages. In this study, researchers engineered a phage's DNA by introducing modified CRISPR-Cas machinery genes. This machinery uses a bacterial immune system as a genome editing tool. With this technology, the scientists precisely modified a bacterial host genome within a miniaturized laboratory microbial ecosystem called EcoFab. Most methods for editing bacterial genomes use circular molecules of DNA called plasmids to transfer DNA between bacterial cells. However, delivering plasmids to specific bacterial species within a mixed microbial community is often inefficient. On the other hand, phages are highly efficient at transferring DNA into specific bacterial species. This research leveraged these phages' properties to develop a new phage-based DNA delivery tool. The researchers also used this tool to precisely edit individual genes inside a target host organism within a living microbial community. Scientists at North Carolina State University developed a single-step phage engineering method that takes advantage of the CRISPR-Cas9 system and a DNA recombination and repair mechanism. Using this method, the researchers engineered the genome of the phage lambda of E. coli. They replaced a non-essential region of the lambda genome with a selectable marker and an engineered base editor. This editor enabled the precise modifications of the bacterial genomic DNA by converting one of the 4 DNA bases or letters A, C, T and G into another. The researchers used this tool to convert a C into a T in specific genes, either within the bacterial chromosome or carried in a DNA plasmid. They demonstrated that the approach was successful by inactivating multiple genes, including the endogenous laxi gene and the plasmid-encoded fluorescent reporter and antibiotic resistant genes. To demonstrate the effectiveness of this precise genome editing tool within a community context, the team used a fabricated ecosystem EcoFab device. Recapitulating a soil microbial ecosystem, they filled the EcoFab with sterile quartz sand and inoculated it with a soil bacterial community composed of E. coli and 2 other known bacterial species. They then added the engineered editor lambda phage that can only infect E. coli to the EcoFab. After incubating the phage with the bacterial community, they managed to base edit 28% of the E. coli cells in the EcoFab. These results highlight the potential of phages as DNA delivery vehicles for targeted members of a mixed soil community for precise genome editing.